No general questions? So I think your quiz is tomorrow, so hopefully you all turn in the lab report already because you probably want time to study. Okay, I'm just going to go ahead and get started. It's being recorded, so people who come late can just watch it. So welcome back, everyone. Remember, tomorrow's your quiz, so make sure you study. We're already past the halfway point. So I've noticed that some people are not turning in maybe a lab. So I'm not sure if a lab will be dropped, but just make sure you turn in all the post labs because they're worth a lot of points. So I don't want you guys to get 0. So I saw that one student didn't turn in one under the last week's post lab. So I'm just making this little announcement for them. So just remember, if you're struggling, come get help. That's what we're here for and all that. Okay, so this experiment is pretty straightforward. There's really no synthesis going on. So not really tricky isolation techniques and stuff like that. So this meeting will probably be pretty short. But let's go straight into today's discussion. Today we're going to learn all about carbohydrates, as you probably have been doing in lecture, especially with sugars, monosaccharides, disaccharides, and going into how they're very important in everyday life, and especially now since we have to tackle, like food shortages problems, things with like maybe replacing meat as our nutrition source. So carbohydrates are going to be an important factor into that discussion. Okay, let's go on. Just remember the post lab for this week due by midnight. Hopefully already turned it in because you need to study. But other than that, no other announcements. So today's learning objectives, we're going to learn about what positive and negative control is or are. We're going to do some enzymatic versus chemical selectivity, and we're going to see why enzymes are pretty hardcore, pretty badass, because they can do the stuff chemistry can do, but under more milder conditions. So they're the rock stars in biology. Okay, green chemistry, we're going to talk about how we can use sugars to make some plastics or make materials out of greener resources. We're going to talk about unknown analysis, and then lastly, you're too bad you can't make it in lab, but you're going to go over how to make a polymer. Okay, so let's talk about the background here. So carbohydrates, they're everywhere. Nutrition, present cell walls, present pretty much everywhere. The most abundant organic compound is cellulose, and that is composed of glucose, so it's a carbohydrate. So they play important roles in cellular metabolism, structure, energy storage, like starch, fats, stuff like that, amylopectin, chitin, and like plants. Also important in other compounds such as RNA, cofactors such as ATP, where we get energy from. So they play many important, they're very prevalent everywhere. So, and they're also the most important present in food and daily nutrition. So that's where food science comes into like kind of melding with chemistry. Okay, so that's the background of carbohydrates. Okay, let's talk about the hydrolysis of starch. Okay, so here are some focus questions, but I don't expect anyone to answer based on how it's gone before. So I'll just go over them after a brief pause. Okay, so why is starch heated in a boiling water bath? Okay, so usually when we do hydrolysis, we need to increase the energy we put in so the activation barrier can be overcome. So we use heat to generate that energy, so generate that kinetic energy. So that's why we need to heat it in a water bath. So that's why using just pure chemicals and no catalyst is unfavorable, because you need to use heat or energy to accomplish this. So we're going to neutralize our solution because we want to stop the reaction, we want to halt it, and we want to prepare it for waste disposal. So if we prepare for waste disposal, we generate less waste, so we make it less hazardous. So that's why when we always do acid-base reactions, we need to neutralize things because we don't want to handle them if they're too acidic or too basic, because that's harmful to us and the environment. So when we're using an acidic solution, we should use a dilute base. So in this case, we're going to use sodium bicarbonate solution or another dilute base, like maybe sodium citrate, sodium carbonate, stuff like that. All right, so the starch test is the starch test. So what is our positive control in this experiment? So let's go over what positive control is. So starch solutions are positive control because it actually produces a positive result, or a result that that comes from a set of experimental circumstances that we should be able to observe. So it is usually part of a test or assay. So if it does not result in a positive outcome, there may be a problem with your assay, your procedure, reagents. So that gives you a good indication whether or not it's working how it should. So that's why you always have some like negative control, positive control, negative, false positive, false negative. So there's all these possibilities when we're talking about science and how it relates to experiments. And this has applications everywhere, right? Like in the health field, positive results for like, like a disease, if you're pregnant, stuff like that. So in chemistry, we do that too. Okay. So so this is a little background about how does the iodine and Ki react with starch, and why does it turn blue? So you can go to this link for more detailed information, but basically there is this charge transfer complex that occurs. So the iodine was not soluble in water, so it reacts with the iodide. They form polyiodide compounds, which results in a very it's a very structured network where it binds to starch and it produces this color. So where does the color come from? Well, the negative charge iodide will donate its electrons as a charge donor, and the iodine will be the acceptor. So we are able to excite these electrons to different energy levels, and the light absorbed that causes that energy change will give you that blue color that we can see with our eyes. So the reason it appears brown in solution is because the absorptions of the different species overlap. So there's a lot of absorptions going on between the iodide and the iodine. So we get a lot of different colors. So once we add the amylose, it forms another complex, and then it acts as a charge donor, the polyiodide as an acceptor, and this complex absorbs at a very different wavelength than the iodine and the iodide, and it turns dark blue. So when we add the amylose there, the starch, that's what causes it to turn blue color. And this is very useful when we're titrating with iodine or iodide. Okay. You don't have to worry too much about that. That's just like a little background information. Okay. So part 2, we're going to hydrolyze sucrose. All right, here's sucrose. So here's a little test question. Hopefully people can answer this. So just gonna open chat. This is a probably important question for a quiz. What are the components or monosaccharides of sucrose? So what are these individual sugars called? You should be able to identify at least sucrose, lactose. I don't know about the other ones. Any ideas? Good. Oh, nope. One's right, fructose. What's the other monosaccharide? Glucose. Good job. So we have glucose here and fructose here. So I don't know if you learned about the cyclic forms yet, but if you were to open these rings up, it'll be glucose and fructose. Okay. So in a previous experiment, we used boiling water to hydrolyze starch, or I think it was starch, yeah, or sucrose. So no, sorry. We did also a hydrolysis experiment, but we used boiling water to break open the sugar, right? Or break open the carbohydrate. In this experiment, we only used an enzyme instead of boiling water. Well, not boiling water, but instead we used an enzyme, no acid, and we perform it at just 45 degrees. So why is that? Well, it turns out that the enzyme is actually a catalyst. So it's actually more efficient catalyst. It's much better than an acid. So it lowers the activation energy. And I don't know if people took biochemistry, but basically the enzyme substrate complex is the intermediate that needs to form for like a reaction in organisms to occur. So enzymes help do this by providing an alternate pathway to form this complex, which then proceeds to product. So that's how enzymes work. Okay. So what does Benedict's test indicate in this experiment? So remember Benedict's test, it will test the presence of a monosaccharide. So the reason it tests the presence of monosaccharides is because we're going to break these up. And a Benedict's test is when you have monosaccharides, they always have a free aldehyde group. And that's why the Benedict's test works, because it oxidizes it to a carboxylic acid. So that's why the Benedict's test is a good assay for the presence of a monosaccharide. Okay. So this is, I think this is the last part of this, the experiment. So we're going to talk about Benedict Solution assay for reducing sugars. I think reducing sugars may be on I don't know if it'll be on the quiz, but it'll probably be part of your post lab. So you probably want to note this. Okay. So what does it test for? It tests for a free aldehyde group, like I said, aka reducing sugar. So a reducing sugar means you have an aldehyde group. If you don't have an aldehyde group, if you can sort of tautomerize to an aldehyde group, like for example, fructose is a ketohexose or ketohexose. So it can tautomerize to the aldehyde, and that's how it becomes a reducing sugar. So that's why it forms a positive test in this assay, as you will find out by watching the video. So all monosaccharides are reducing sugars because they have a free aldehyde group or they can convert to one. So what must occur for a positive Benedict's test? So if you read in the lab manual, the positive test is we need both a color change and a precipitate. If you don't have a color change, if you only have one or the other, that's not going to work. That's what we call a false positive. So in the video, be on the lookout for that. Students who did this lab before, they had trouble deciding what one change meant and not the other. So you need both of these things to be a positive test. Okay, so let's talk about the reaction. So what is being reduced and what's being oxidized? So we talked about sugars having free aldehyde groups. So that's the thing that's going to be part of this reaction. So oxidized is our sugar. Reduced, so the blue solution in Benedict's is caused by copper 2. So that's what's being reduced because we're forming a copper oxide. So see here, we form a copper here. It's becoming copper 1+, copper 2+, the 1+, that is reduction. Sugar here is coming from an aldehyde. It will react like this in this fashion to produce a carboxylate. So it's being oxidized because we're adding oxygen in place of a hydrogen here. So that's called oxidation. So think about if I go from hydrogen to oxygen, oxidation, oxygen. Okay, then we get red precipitate, so like a brick red, like brick powder, not soluble in anything, not even water, and we produce water at the end because it's an acid-base reaction too. to balance out the H here. Okay, so that is Benedict Solution for reducing sugar. So that'll probably be the last part of the experiment. And I believe that is it. Let me double check. Oh no, not yet. Sorry. Okay, lastly, we're going to talk about synthesizing a biopolymer from potato starch. So you take starch, from the grocery store, and we're going to make a biopolymer out of it. So we're going to use a polymer. We're going to make a polymer by using borax, and borax basically is boron with 4 oxygen bonds, and it'll form covalent linkages or strong covalent bonds between the layers of starch. Too bad you can't make it. So what this does, this strong cross-linking causes this to make the polymer feel like slime or silly putty. As a kid you play with silly putty. You can stretch it. It won't be brittle. It won't break. You can tear it apart. It's not like Play-Doh though. It's like putty, not Play-Doh. Yeah. I played with that as a kid. I don't know if kids like that now. They probably like slime stuff, slime videos. Anyway, so the borax is actually it's to create create these bonds between the layers of starch, and that creates your polymer. So that creates the strength or the puttiness of your polymer. So that gives it its stretchy character. Okay. So borax, the reaction is endothermic because you end up needing to heat it to get the reaction to go. So it's not exothermic. So if we're making a polymer, we want to extract it. We want to isolate it. So we need to neutralize the reaction to make it safe. To make it safe to touch and stable. Because if you were to make this, you would, at the end of the day, you could go ahead and try and stretch it. And it was a pretty cool experiment. Unfortunately, we can't do that here, but this is how it applies to making polymers, because polymers, they make very useful materials that we use in everyday life, so they have many applications. So everyone like polymers are always things that people look for, or maybe like for research. So it has many applications. Okay. So what are the benefits of bioplastic? So anyone like cares about climate change? We need to kind of replace materials. We need to kind of find a way to recycle and recycle everything that goes into making plastic. So we can't just take stuff from oil and make that into plastic, because we can't replace the oil. The oil turns into CO2. So we can't really replace it in a cycle. So the benefits to using bioplastic is that starch, it can be broken down by enzymes, so it's very biodegradable. So what we call this is it has an enzymatic breakdown. It breaks down in the body. So pay attention to this point right here, because I don't know if anyone, not many people come to office hours, and this will be important for one of the post-lab questions. So I would pay attention to this part right here. And it's sustainable feedstocks. Starch, you can extract it from plants and stuff like that. So it's always it's in the cycle of an organism. So that's what we call renewable, because it has a cycle of being produced, and we can consume it. And there's always some other source that produces it naturally, and that's how we get a renewable feedstock or source of a organic compound. Okay, so that's how you make the potato starch biopolymer. And yeah, that is it. All right, if there ain't no more questions, I'll see you guys next week, and good luck on your quiz.